Science is often taught as a series of breakthroughs. One discovery, one name, one moment that changes everything. A lone genius, a sudden insight, a clean turning point. This is how progress is remembered, but it is rarely how progress actually happens. Scientific discovery is slow, messy, collaborative. It depends on observation, translation, repetition, and interpretation, and the people who perform those roles do not always become the ones remembered. The great scientist narrative is appealing. It simplifies complexity. It gives history a hero. But real science doesn't move forward through isolated brilliance. It advances through systems. Experiments are designed carefully. Data was collected patiently. Patterns recognized over time. Ideas translated into shared language. Yet when history is written, credit often collapses inward toward the most visible figure, not always the most important one. For much of scientific history, access mattered as much as insight. Who could publish, who could lecture, who could hold institutional positions. Those constraints shaped not just who did science, but who was recorded as doing it. Many contributors worked in supporting roles, some by necessity, some by exclusion, and some of the most foundational scientific advances came from people whose names never became headlines. Modern chemistry is often associated with Antoine Lavoisier, but his work depended heavily on the contributions of his wife and chief collaborator, Marie-Anne Pause Lavoisier. She translated scientific texts into French. She standardized terminology. She created precise illustrations of experimental apparatus. These were not secondary tasks. They were how chemistry became shareable, repeatable, understandable. Without translation and structure, experiments remain isolated events. With them, they become science. In the mid-20th century, understanding the structure of DNA required more than theory. It required data. That data came from X-ray diffraction images produced by Rosalind Franklin. Her images revealed the helical structure of DNA, but she did not control how that data was interpreted. Others built the model. Others presented the conclusion. History remembered the theory, not the measurement that made it possible. This was not theft in a dramatic sense. It was a system where interpretation carried more weight than generation. Astronomy offers one of the clearest examples of how invisible work becomes foundational. At Harvard Observatory, a group of women known as the computers cataloged and classified stars. Among them was Henrietta Swan Leavitt. She discovered the relationship between a star's brightness and its distance. This single insight made it possible to measure the scale of the universe. Without her work, cosmology as we know it does not exist. Yet her role was analytical, systemic, quiet, and history tends to overlook quiet revolutions. Scientific progress often depends on collaboration. But recognition depends on ownership. Who presents the findings? Who publishes first? Who speaks for the data? For much of history, those opportunities were unevenly distributed. This does not make science corrupt. It makes it human. The story that survives is often the story that fits the structure of its time. Long before genetics became a field, patterns of inheritance were being studied. Nettie Stevens independently identified the role of sex chromosomes in determining biological sex. Her work laid the foundations that modern genetics still relies on. But early genetics did not yet have the prestige or structure to elevate all contributors equally. Some discoveries entered textbooks, others became footnotes. Some contributions are overlooked not because they are minor, but because they do not fit the heroic narrative. Cataloging, classification, measurement, translation. These are the scaffolding of science. Without them, breakthroughs collapse. Yet history tends to reward conclusions over process. This pattern is not confined to one era. or one group. It emerges whenever systems prioritize visibility over infrastructure. Science advances through networks. History remembers individuals. The tension between those two forces shapes what we inherit as truth. Modern science is more collaborative than ever. Large teams, shared datasets, distributed discovery. Yet the instinct to simplify remains. We still look for singular figures, clear authors, clean stories. Understanding how science actually advances helps us see progress more honestly. It reminds us that discovery is collective, and that forgetting process means misunderstanding how knowledge is built. This is not about correcting history to assign blame. It is about widening the lens, seeing science not as a sequence of geniuses, but as an evolving system built by many hands, some visible, many not. When we simplify discovery, we lose something important. Not just names, but understanding. Science is not a miracle performed by individuals. It is a structure sustained by collaboration. Remembering that does not diminish progress. It explains it. Thank you for watching. Join us every Sunday for deeper stories, bigger questions, and long-form explorations that go beyond the short videos, because knowledge doesn't stop at 60 seconds.
Thanks for watching. This is 60-second knowledge now extended, because curiosity doesn't stop at 60 seconds. Subscribe on YouTube and follow us on TikTok.